Okay. Very good morning to all of you and welcome to IFAS. Welcome to IFAS, guys. Good morning. Very good morning, everyone. Hope all of you are doing very well. And I also hope that you are very eager to learn new things in bioinorganic chemistry, because bioinorganic chemistry is a very interesting chapter. And now, up till we have discussed two lectures of bioinorganic chemistry. In that we have seen basic segment, okay, basic things regarding bioinorganic chemistry, as well as we have seen Heme Unit, okay. So up till now you might have got the idea that bioinorganic is going to be one of the most interesting topic that you can ever learn, okay. So very good morning, okay. Cool Bhilai, good morning sir, very good morning. Cool Komal Sharma, very good morning. Mamta, good morning. Sumitra, good morning, Sirji. Very good morning, Sumitra. Bhumi, good morning, sir. Very good morning. Good morning, everyone. Yes. Okay. Quickly join the session, guys. Okay. Quickly join the session. And first of all, okay, whenever you join the session, make sure you hit the like button. Okay. Because we can understand, okay, we will get the idea that you enjoy our session, and it motivates us to keep on going, to keep on improving ourselves and give you more wonderful content that you can ever provide, okay. So like the session as well as share it with your friends, because if you care about us, you will like the session, but if you care about your friends, you will share this session with them, okay. And if you are new to the channel, make sure you subscribe the channel, because whenever any exam related notifications or news you might want to know, okay, that will be available for you, okay. You don't have to worry about it, okay. Okay, good morning, very good morning, everyone. So let us start the session, okay. But before starting our today's topic, okay, we are going to today discuss regarding heme unit as well as non-heme unit, okay. Basically we are going to discuss non-heme units such as hemocyanin as well as hemerythrin, okay. We are going to talk about it, okay. But revision is must and necessary, okay. Revision is must and necessary, okay. So let us revise what we have learnt in our last class, okay. We'll have a look at a basic portion, okay. We'll look at the basic portion of inorganic chemistry that we have discussed in the last class, okay. Sinu Pandey, good morning sir, very good morning Sinu, how are you? And hope all of you are doing very good in your life as well as in your preparation, okay. And hope this session also helps you in preparing well for the exam, okay. Okay, so let us start the session, guys. In today's session, what we are going to have a look at is non-heme unit as well as hemerythrin as well as hemocyanin, okay. These are the things that we are going to focus on, okay. But first, okay, very good news, a very wonderful thing that I would like to share with all of you is around 1200 plus IFASians have qualified CSIR UGC NET examination only in the December session, okay, only in the December session, as you can see here, okay. So it is a very proud moment as well as wonderful moment for us, and I would like to share it with all of you, okay. It makes me very happy, okay. Okay, now let's move on, okay. Now let us talk about the things that we have seen in the last class, okay. Now in the last class we have majorly talked about hemoglobin as well as myoglobin, okay. So now let us understand, okay. Now we have discussed the structural features, okay. We have talked about iron as well as their high spin, low spin conditions in deoxy form, oxy form, dome state, T state, R state. These were the things that we are talking about in the last class. But in today's class, let us have a different perspective. Let us have a clear understanding. What, sir? What clear understanding that we will have here? Yes. What is that we should learn now? Okay. We should learn regarding the function, how they work in a synchronized manner. Okay, it is very important that hemoglobin as well as myoglobin, they work in a synchronized manner. Okay, if they not, then there will be a lot of trouble for us. Okay, so let us understand. Okay, so now let us understand this process here, right now, right here. Okay, so when we inhale, okay, all of you inhale. Okay, I inhaled air, okay, air through my nose. Okay, in that air you will find a mixture of gases. NO2 is present, CO2 present, SO2 is present, N2 is present, majorly is N2. But in that a very important component is present, that is what? That is O2, sir, oxygen, है ना? It is necessary for life, it is essential for life to carry on. Okay, so in that mixture of gases, oxygen is present. Okay, so I will focus only on oxygen. Sir, what about other gases? Okay, I don't care about those gases. Okay, right now they are not of my concern. Okay, my concern is to understand the oxygen transport that is required to produce energy. Okay, so I will only focus on that part. Okay, Swati, Prerna, Diksha, Pragati, good morning all of you. Very good morning. Hope all of you are doing well. Okay, and to get inform you about this, we were discussing the function of hemoglobin and myoglobin. Okay, so now what we were talking that when I inhale, okay, when I inhale there are a lot of mixture of gases, but in that oxygen is important for us, so that we will only focus on oxygen. Okay, so now when I inhale, oxygen enters in my body. Okay, and to be specific, oxygen enters where? Oxygen enters lungs. If you are a non-aquatic animal, if you are a aquatic animal, oxygen will enter where? In the gills of the system. Okay, in the gills of the species. Okay, because aquatic animals breathe through gills. Okay, gills are important for them. Okay, just like how lungs are important for us. Okay, so right now when I inhale oxygen, this oxygen enters into my lungs. Okay, then there HB is waiting. Okay, hemoglobin is waiting. Okay, it is seeing its clock, watch. It's saying that the human is taking more time to breathe. Okay, if you stop breathing, the first concerned species will be hemoglobin. Hemoglobin will think what he is doing. Okay, is why he is not breathing. Okay, it is very important for hemoglobin that you breathe, so that hemoglobin can carry out its function. Okay, hemoglobin is basically waiting in lungs. Okay, lungs or exterior part of blood to pick up the oxygen, because what is the function of hemoglobin? Yes, what we have discussed is hemoglobin carries oxygen, transfers oxygen where it is necessary. Okay, so whenever you take oxygen through lungs, hemoglobin is waiting there, and when the instance, okay, at the instance when oxygen is present, hemoglobin carries the oxygen. Okay, hemoglobin carries the oxygen to the muscle part of the tissue. Okay, to the muscles. Okay, to the muscles. Now, in muscles, what is present? In the muscles, myoglobin is present. Okay, in the muscles, myoglobin is present there. He is also waiting. He is also looking at its watch. Okay, he is saying hemoglobin is taking much of the time. Okay, and he might also imagine that it is stuck in traffic. Yes, it's stuck in traffic, but the wait is now over. Okay, as soon as hemoglobin arrives at the center of action, okay, at muscles, then myoglobin is the one that picks up the O2. Okay, now what is happening? Yes, this is you. Okay, this is you. Okay, so now you are inhaling oxygen. Okay, I will draw this somewhat bigger. Okay, this is you. Okay, you have eyes, you have nose. Okay, so now whenever you inhale oxygen, okay, what happens? It travels to the lungs. Okay, it travels to lungs, and there hemoglobin carries out the oxygen, and that oxygen is carried by myoglobin, or that oxygen is carried to myoglobin, where in the muscle part, okay, in the muscle part of tissue. Okay, so now what it does is, this myoglobin, which is storing the oxygen, it will store oxygen. Sir, why it will store oxygen? What is the concern behind storing the oxygen? The concern is very simple. Okay, what plants are doing? Okay, plants are synthesizing C6H12O6. Okay, what are plants doing, sir? This is ground, this is a tree. Okay, let us suppose this is tree. Okay, please understand this. Please understand this very clearly. Okay, you might understand the importance of plants here as well. Okay, so now what does this plant do? This plant takes the CO2 that you are exhaling. This plant exhales the CO2. Or let us try to come to the first plant. Okay, this plant exhales oxygen. This oxygen is taken by you. This is transferred to the lungs, then hemoglobin, then muscle tissues where myoglobin is present, where it stores the O2. Okay, then what happens, Sirji, here? Yes, what you will do is, whenever you are exercising, when you are studying, when you are doing nothing, you require energy, and that energy is coming from where? Is coming from burning the C6H12O6. Okay, that is a form of energy, glucose. Okay, so here O2 is required, sir, burning. Yes, you might have learned this in your school days, that oxygen is very necessary to burn things. Yes, oxygen is very necessary to burn the things. So therefore, to burn C6H12O6, what you need is, you need oxygen, and with the help of oxygen, what you will do? You will produce the most important content, that is energy. Yes, you will produce energy, that is the fuel to do work, to do nothing, to study, to sleep, to do everything. You require energy. Okay, now there are some byproducts along it. Okay, there are some byproducts along it. You will produce carbon dioxide. You will produce what? You will produce carbon dioxide. Okay, you will produce carbon dioxide, as well as you will produce water. Okay, as well as you will produce water. So for the humans, water is not of much concern, because water is utilized in the system and it will be eliminated through various processes, like its sweat, like urination. Water is eliminated. But what we will do with the CO2, sir? What is the function? What is its usefulness? Yes, CO2. Yes, CO2 is not useful for us. Okay, CO2 is not useful for us. So therefore, what happens is, CO2 is exhaled. I inhaled air or oxygen. All this process happened here, and when I exhale, CO2 is eliminated. Okay, CO2 is eliminated. See how interesting and wonderful this process is. Okay, so within an instance, these process occurs number of times. Okay, within an instance, within a second, these process can happen number of times. Okay, as the number of inhale exhalation happens, these all process are carried out. Okay, so now this CO2 is exhaled. I exhaled, and CO2 was exhaled through this exhalation, and this CO2 was taken by your tree or plant. Okay, this CO2 was taken by this tree or plant, where what it does is, it does the opposite thing that you do. Yes, what it does is, it exactly does the opposite thing that you do. Okay, okay, it's not visible. Okay, let me make it visible for you. There is no problem in that. Okay, okay, so hopefully now this is clear, visible. Okay, so see, you took oxygen. Yes, with the help of oxygen you burnt the energy source, and with the burning the energy source, CO2 was released. Now this CO2 is not useful for us. Then you will exhale it, and this exhaled CO2 directly goes to a plant, where what it does is, it does the exact opposite of this reaction. What it does is, it does the exact opposite of this reaction, where it utilizes the 6 CO2 and where it requires H2O, and this energy comes from where? From sunlight. Yes, from the source of very life, that is sun. Okay, so this reverse reactions takes place. Okay, with these all things, C6H12O6 is synthesized along with oxygen, and then again this oxygen is taken by whom? Taken by this person, and then all this process happens. Then again CO2 is released, then again this plant or tree takes the CO2, and through CO2 taken, this opposite reaction is happening. Okay, so you can understand this, how synchronously this process is happening. Okay, and now let us come to the human part. Okay, let us come to the human part. Now what happened here? Now you produced CO2. Okay, it's not like that you exhale CO2 will directly go. Okay, there is some necessary component that is required to travel or transfer the CO2. Okay, first of all, this CO2 that is not necessary in my body, it's converted into what? It is converted into HCO3. Okay, you can see here the reaction, CO2 along with H2O, that gets converted into H2CO3. Okay, and this is carried out by a very important catalyst. Okay, I am calling this as catalyst. I am not telling it as enzyme or anything, because basically enzyme is also a catalyst. Okay, you can call this as biocatalyst. Okay, you can call this as biocatalyst. So now this biocatalyst, what it does is, it converts CO2 into H2CO3, and later this is converted into HCO3 negative. Now this HCO3 negative is transferred into the blood serum, where through this it travels to the lungs, and through traveling to the lungs it is getting converted into H2CO3, and at low pressure H2CO3 gets converted once again to CO2, and when you exhale, CO2 is eliminated through your body. Okay, yes, see how wonderful this process is. Okay, okay, so hope all of you have understood this process. Okay, tell me whether it is clear. Yes, tell me whether every one of you have understood this process. Yes, please let me know in the comment box, is there any doubt, and if they are, feel free to ask. Okay, there is nothing to worry about. Okay, Anju is saying yes. Okay, very great, good. Yes sir, Preeti. Okay, fine. Okay, if you have any concern, okay, please let me know in the comment box. Okay, so now let us move forward. Okay, so this is the picture that we were discussing yesterday. Okay, it seems very simple when you look at it in a simpler manner, but when you focus on the details, it is very complicated. But the magic lies in the complication. Okay, the magic lies in the complication, because here you can see, yes, this iron is what? This iron is above the plane. This iron is where? Above the plane. Okay, and this is called as T state, or you can call this as dome structure also. You can see here in this image. Okay, so here this is dome state or T state. Okay, so whenever it attaches with the oxygen, okay, what happens? Its size decreases, and it slowly, what happens? It slowly gets along with the plane. It slowly gets along with the plane, and it will look like this. Okay, here it will be present here. Okay, oxygen when it combines with oxygen. Okay, so this is how a very phenomenous manner oxygen transport and oxygen carriation or storage happens. Okay, and one more image that I would like to show is more complicated structure of your hemoglobin as well as myoglobin. Now understand this part very well, okay, because on the basis of what we are going to discuss, a question was asked in GATE, okay, you have been asked this in your GATE examination, okay. Now you can see here guys, okay, now you can see here there are two types of histidine, okay. Here there is oxygen, okay, this is my oxyhemoglobin or oxymyoglobin, okay. See here there is boundation of O2 with iron, okay. Now this oxygen is hydrogen bonded, this oxygen is hydrogen bonded through histidine, okay. So now this type of histidine is called what? This type of histidine is called distal histidine, okay. This type of histidine is called as distal histidine. Remember this point very well, because this has been asked in your previous GATE sessions, okay. And one more histidine that was present was at 5th coordination site of this iron, okay. This is 5th coordination site, and there also lies a histidine, okay. So now this type of histidine is called as proximal histidine, okay. This type of histidine is called as proximal, okay. The iron, the histidine below the iron, the histidine below the iron, the histidine below the iron is called as proximal histidine, and the histidine above the iron is called as distal, okay. So repeat this few times and you will memorize it and you will get comfortable with it, okay. So what is the function of your distal histidine, okay? In that, distal histidine is important. Can any one of you tell why it is important? Yes, I want to see all of you answer this, okay. I want to see all of you answer this, whether it might be correct, whether it might be wrong, it doesn't matter for us, okay. It is not a matter of concern. What is the matter of concern is you are willing to answer, that is important, okay. So please try, okay. Can you tell me why distal histidine is important? What is its function? Yes, please comment as soon as possible. Yes, what is the function of distal histidine, guys? Okay, please let me know in the comment box, okay. Quickly, okay, quickly respond all of you. Markande Jaiswal, good morning Sirji, very good morning Markande. Can you tell me about the function of distal histidine, Markande? Preeti, Pragati, Diksha, Anju, Mamta. Chem Pacific has also joined the session, okay. Subha Sakal, आदरणीय प्रणाम गुरुजी, कसं आहे तुम्ही? जी pranam Chem Pacific. I am doing fine and hope you are also doing fine, okay. Chem Pacific, can you tell me the function of distal histidine? Okay, skeletal formation, no, okay. Anju, okay, skeletal formation, no, okay. I think it's quite tough for you to answer this question, okay. Just let me answer it for you. Okay, I am seeing answers. Mamta, very good. See, Mamta has said that bent form में convert करता है. That means it converts it into bent form. Okay, very good. Markandeya Jaiswal, amide group. Okay. Carbonyl poisoning नहीं होने देता. बहुत good. Very good, Chem Pacific. Diksha, distal histidine bind with O2. Preeti is saying that because oxygen binding with iron. Very good, guys. Very good. Your effort. Okay, these are your efforts, and I can clearly see that you are putting a lot of effort to develop, to enhance your knowledge, to develop your knowledge, and it is very good. Okay, so keep improving, keep putting efforts into. Okay, because effort will not go in vain. Okay, effort always helps you in your lame situations. Okay, okay. So the most important comment that I have got here is of Chem Pacific. Okay, see, now when you travel, okay, nowadays lot of pollution is happening. Okay, because of vehicle pollution, industrial pollution, etc, etc, etc. Okay, so these days there is a lot of cause of pollution. Okay, so in this pollution, a very important component of pollutant is carbonyl monoxide, which we have not discussed up till now. Okay, so let us try to get the culprit out of the picture. Okay, in the picture actually. So carbonyl, yes, what it is? Carbon monoxide. Carbon monoxide is a very dangerous gas. Yes, carbon monoxide is a very dangerous gas. Why? Because it is very dangerous because it binds with a greater affinity than oxygen. Okay, if you take oxygen, if you take carbon monoxide is the one that is having greater affinity towards your hemoglobin. So whenever your hemoglobin is present, oxygen is present, carbon monoxide present. If carbon monoxide binds with the hemoglobin, it cannot detach. Yes, it cannot detach. But if we take oxygen, yes, oxygen, if it binds with your hemoglobin, it attaches, it detaches very smoothly. Okay, because it is a pH dependent species. Okay, and one more thing here important, what is important for us is the nature of ligand. Okay, here if you take carbonyl and O2, now just tell me which is a greater pi acceptor. Yes, which is a greater pi acceptor here, guys? Greater or good pi acceptor? Yes, which is a good pi acceptor? Yes, very good, Campus Vic. And all of you, Silky Agarwal, Anju, very good. Okay, so carbonyl is a greater pi acceptor. Okay, so whenever here carbonyl comes, yes, what it does is it binds it in a linear manner. Yes, it binds it in a linear manner where its orbital can accept pi electron density. So therefore a greater bond strength is happening, greater back bonding is happening. So for this reason, what happens now, when it binds, it cannot detach then. Okay, it cannot detach then because of greater pi acceptance. Okay, so therefore here comes in the picture distal histidine. Here comes into the action what? Distal histidine. Okay, so if you look at distal histidine, it forces, okay, what it does is it forces the carbonyl to bind in a bent manner, so that the orbital is present like this. So lesser amount of overlapping takes place. Okay, and the bond that is being formed is having lesser strength, then it can dissociate easily. Okay, so therefore distal histidine prevents hemoglobin from carbonyl poisoning. Okay, prevents hemoglobin from carbonyl poisoning. Okay, remember this point. Okay, all of you got this? Yes, tell me whether it is clear. Tell me whether it is clear or not. And if you are having any doubts, please let me know, guys. Yes, comment it. If you are having any doubts, let me know. Okay, okay, great. So let us move on. So now let us talk about non-heme units. Okay, so let us talk about non-heme units. Okay, so there are basically two types of non-heme units. First one is Hemerythrin and the second one is Hemocyanin. Okay, yes, so let us talk about these non-heme units. Okay, but first, all of you tell me what does non-heme unit means. Okay, can all of you define what is non-heme unit? Yes, whether if you are able to tell what is non-heme unit. Yes, very good, Campus Vic. Okay, yes, Can all of you tell me what does non-heme unit means? Okay, the first one to comment the answer, guys. The first one is Silky Agarwal. No porphyrin ring. Very good Silky. Anju, heme group not present. Very good. Campeswick, non-porphyrin ligand present. Very good. Yes. See, same thing. You can explain it in different manners. That is very important. Okay. Diksha, porphyrin ring not present. Very good. Campeswick, absence of heme. PKP, no Fe present. No. Yes, this is not a proper definition PKP, but don't worry. Okay. Bhumi, absence of porphyrin. Very good. Okay. So let me tell you what does non-heme unit means. Okay. Simply here porphyrin ring. Porphyrin ring is absent. Okay. Porphyrin ring is absent. Okay. So there are two types of proteins that we are going to discuss. The first one is Hemerythrin and the second one is going to be Hemocyanin. Second one is going to be Hemocyanin. Okay. So let us discuss them one by one. So first we are going to discuss is Hemerythrin. Hemerythrin. Okay. Let us first discuss Hemerythrin. Okay. So if we talk about Hemerythrin, there is a major metal that is present. Okay. A major active site that is present is which metal? Yes, it is having iron. It contains Fe atom. It contains iron atom. Okay. It contains what? Iron atom. Very good, guys. I can see that. Yes. This fills me with a lot of confidence. Okay, this actually fills me with a lot of confidence that you guys are understanding what I am trying to tell. Okay, that is very good. Okay, so second point regarding Hemerythrin. Okay, it does not have heme unit. It does not have heme unit. Okay, because Hemerythrin does not contain porphyrin ring. Okay. So then what is present here, sir? Yes. How it functions? Okay. First of all, the function of Hemerythrin, it is transportation of oxygen. It is transportation of oxygen. Okay. So now in humans you will find hemoglobin, myoglobin, etc. like these species. But where Hemerythrin is present? These are present in invertebrate, guys. Can any one of you now define what is invertebrate means? Yes, can any one of you define me what does invertebrate means? Okay. So it is very astonishing that we come across, see, the content is very simple, but see the things that we are covering here. Okay. So now you will understand what does invertebrate mean. Okay. So let me know if you know invertebrate, vertebrate. I think you have learned this in your school days. Remember your school days? Yes. School days are of a different era. They are of having different emotion. Okay. Where we used to play, we get scolded from the teachers. We got beaten also sometimes. Okay. We used to have a lot of fun. Okay. No. I am not asking about examples. I am asking what does invertebrate means. Yes. Very good, Sabhya, sorry. Sabhya, very good. Yes. Very good. Anju, very good. Okay. So, invertebrate, what does it means? Absence of backbone. Yes, nerve cord not present. This is called invertebrate. They are called invertebrate. Absence of spinal cord. Very good, Muhammad. Without backbone. Yes. And vertebrate means with backbone. Okay. So now vertebrates have, basically vertebrates or mammals I can say, they are having hemoglobin as well as myoglobin which are heme unit. Okay, but what about invertebrates, sir? What about octopus? What about mollusks? What about snail? What about arthropods? What we talk about them? How they transport the oxygen in them? Yes, for them there is presence of hemerythrin. Okay, so there is hemerythrin that is present. Okay, so hemerythrin is found in where? In invertebrates, like for example snail, octopus, arthropods, mollusks. Okay, yes. Okay, so let us have a closer look at the structure. Okay, let us have a closer look at the structure. Fe is present in +2 oxidation state and each active site carries 2 Fe. Earlier you have seen hemoglobin as well as myoglobin, there will you find only one iron, but here there is a requirement of 2 irons. Okay, these 2 irons are connected with each other through oxygens of aspartate. Okay, and one more species that is present is glutamate. Okay, glutamate, and there is also a hydroxy bridging present. Yes, there is also a hydroxy bridging present in your hemerythrin. And not every, this iron, okay, let me talk about this iron. This iron is connected with 3 histidines, nitrogen. Okay, nitrogen of 3 histidine. But the second iron, if you see here, it is only connected with 2 histidines. And this is very important. You might think this is a major concern for us. Okay, there is a manufacturing default here, but no, here it is actually very sensible to have one iron which is having 3 histidine and the second iron which is having only 2. Why? Because whenever the oxygen comes, okay, whenever the oxygen comes, what happens here? Yes, so this was your structure. Both the iron were in +2 oxidation state earlier and these were connected with the hydroxy group as well as aspartate as well as glutamate. Okay, so when oxygen binds at the active site, how this oxygen binds here? Yes, this is the oxygen. Here oxygen binds in this fashion and there is interaction between this hydrogen as well. There is interaction between this hydrogen, or I can say hydrogen bonding is present. Okay, so here you can see this iron which was having 3 histidine it was hexa coordinated. Okay, it was hexa coordinated, but now you can see this penta coordinated which were having 2 histidine only. Now this is the site where oxygen binding happens and there is hydrogen bonding as well as with the hydroxyl group. Okay, with the hydroxyl group, so that both of these become hexa coordinated. Both of these become hexa coordinated. Okay, where earlier you can see this is hexa coordinated, but this iron is what? Penta coordinated. This iron is what? Penta coordinated. Okay, so this is the major difference which actually helps the oxygen to bind with hemerythrin. Okay, and this is called as deoxy hemerythrin and this is called as oxy hemerythrin. This is called as oxy hemerythrin. Okay, so I think you get my point. Okay, one more thing. Very good, Anju. Yes, very good, Preeti. Very good. Okay, one more thing that we should change here is the oxidation state. Yes, Anju. Yes, I got your point. Thank you for reminding me about the oxidation state of iron. Okay, so what will be the oxidation state of iron now? Earlier they were in +2 oxidation state, but now as soon as oxygen binds, these iron gets converted into +3 oxidation state, 1, 2 and 3. Okay, so these iron are present in now +3 oxidation state. Okay, okay, so let's move forward, guys. Okay, I hope everything is clear until now. Okay, so let us talk about their differences. Okay, let us talk about their differences. Okay, so let us talk about their differences. Here I will take deoxy form and here we will take oxy form. Okay, so the first thing about this deoxy form here is both iron, both Fe are in +2 oxidation state. Can any one of you tell me whether they are in high spin state or in low spin state? Yes, can any one of you tell me whether they are in high spin or whether they are in low spin state? Oxyhemerythrin is red colored due to LMCT, but hemerythrin is colorless cause diamagnetic. Very good campus week. Yes, very good guys. Campus week, Anju, PKP, Preeti, Mamta, the moon, very good morning to you all. Okay, I had seen but I forgot to reply to your comment. Okay, very good morning. Diksha, yes, very good. Campus week, Afrin, very good. Okay, so here you can see that both the iron are present in high spin state, but as soon as they get converted into oxyform, these iron gets what? They also get converted into +3 oxidation state. Yes, two things happen here. The iron gets converted into +3 oxidation state and the spin state also changes. From high spin they are getting converted to low spin. Okay. Second point, sir. Yes, here you can see the first iron was what? Hexa coordinated. Yes, it was having coordination number 6. While the second iron in deoxy form was penta coordinated. It was penta coordinated. Okay. Yes. But if we talk about oxyform, what happens here, sir? Yes, if we see oxyform here, what happens? Both the iron, both Fe are in hexa coordination. Both Fe are in hexa coordination. That is, they are having coordination number 6. Okay, they are having what? Coordination number 6. Okay, third point, guys. Okay, let us discuss regarding their magnetic property as well as the number of unpaired electrons. Okay, so if we take iron, okay, it is in +2. +2 means it is having D6 configuration. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Okay, so these are the splitting of iron. Okay, in exactly octahedral splitting happens here. Okay, so they are having 6 electrons which are filled in high spin state. 1, 2, 3, 4, 5 and 6. Okay, so by seeing 4 unpaired electrons, you might think, sir, this is going to be paramagnetic. It is very obvious. But you have, then if you think like this, you have missed the larger picture. Okay, the larger picture, there is a twist. Okay, in this story there is a very large twist. Okay, it is very interesting. Okay, so now these 4 electrons, okay, these 4 electrons and these 4 electrons, what they will do is, yes, they will coordinate with each other, they will talk with each other, they will interact with each other, and they will tell that why not we couple with each other. Yes, why not? Yes, why not? So they couple with each other, and through this process coupling happens, and this is called as antiferromagnetic spin coupling. Antiferromagnetic spin coupling. Okay, and due to this spin coupling, okay, tell me whether it is paramagnetic or it is diamagnetic in nature. Yes, can you answer this question, whether it will be diamagnetic or paramagnetic? Okay, in my thoughts I think it is a very simple question. Okay, but it is very simple for me. Okay, it might be simple for you or it cannot be. Okay, but by looking at your answers, guys, okay, by looking at your answers, I got confirmed that it was a very simple question. It was actually a very silly questions for you. Okay. Yes, basic. It is very obvious that this is going to be diamagnetic. Okay, this is going to be diamagnetic in nature. Okay. So let us talk about oxyform. What happens here in oxyform? Now both the iron were in +3 oxidation state and in low spin. Okay, so this is the splitting, crystal field splitting. Okay, I have how many electrons? 5 electrons. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Okay, yes, very good guys. So here if you see this one electron and this one electron, they will try to interact and couple with each other, although they are very far away. Okay, although they are very far away, they will get connected. Okay, through the process called antiferromagnetic spin coupling. Through the process called antiferromagnetic spin coupling, and through this spin coupling, this also makes the species diamagnetic. This also makes the species diamagnetic guys. Okay, okay, very good guys, very good. Okay, so let's discuss few more differences that are going to be important. Okay, so now point number 4, let us talk about ESR activity. So tell me in the comment box whether they are ESR active or whether they are ESR inactive. Yes, whether they are ESR active or whether they are ESR inactive. Yes, both of these are ESR inactive. Okay, and this also oxy species, whether it be oxy, whether it be deoxy, these both are ESR inactive. Okay, fifth point guys. Okay, because of diamagnetic property, this is going to be colorless. Because of diamagnetic property, this is going to be colorless. Okay, but the oxyform, okay, but the oxyform, it is what? It is red in color. You might think why it is red, sir? Yes, it is also in diamagnetic nature. Okay, so then why it is red? This is because of a phenomena which we learn in coordination chemistry, which we learn in spectral properties of coordination compound or other species. Okay, that is called as ligand to metal charge transfer. Okay, sir, what is the ligand present here? Arey, it is oxygen guys. Yes, see oxygen. Yes, you can see here oxygen. Okay, this is your ligand, and here you are having metal. Okay, so through charge transfer, and this basically is called ligand to metal charge transfer. This is red in color. Okay, so now one more very important point, that is regarding O2 bindation. O2 binding. Okay, O2, how it is bind? Okay, it is bind in the form of what? HO2 negative. It is bound in the form of HO2 negative. Okay, where its stretching frequency, what happens? Its stretching frequency decreases further. Okay, now this is very important part guys. Okay, this point you should remember, it is around 800 to 850 per centimeter. It is around 800 to 850 per centimeter. Okay, yes, tell me whether all of these points are clear for you. Yes, we can say it as peroxide. Okay, so just let me know whether everything that we have discussed till now is clear or not. Very good, Preeti, the moon, Anju, Camp Pacific, Diksha, Mamta, Mohammed, the moon, Bhumi, very good guys, Afrin, Ekdam chakachak chamka. Okay, very happy to see this. Okay, so now one thing I would like, okay, one thing I would like to add here is regarding IFAS app as well as its website. This website will be very beneficial while you are preparing okay, because here you will find lots of resources that you can come across and that you can get help from, okay, while you are preparing for CSR NET examination or else any exam you say about, okay. You will find sources that are related to those exams, okay, like PYQs, paper analysis, cutoffs, and many more things, okay. Just log into www.ifasonline.com, okay. And if you are preparing for CSR NET examination, whether it might be in the June session or in the upcoming December session, okay, you can enroll in our CSR NET 2024 target batch, okay, that too at flat 75% off, okay, by using the promo code IFAS75, okay, by using the promo code IFAS75, okay. Radiate brilliance, okay. I find it very ironic because in Bark, okay, we study nuclear chemistry or nuclear sort of thing where radiations are present. So here radiate word is used, okay, and it is very relating, okay, it is very correlating. There radiations are emitted, here you will radiate brilliance, okay. So through this recorded course you can radiate your brilliance, not that you will emit gamma rays, not that you will emit beta alpha rays, okay. You are not radioactive, might be, okay, I don't know, okay. Okay, so here you can use the promo code IFAS75 and get flat 75% off on all the courses, okay, basically which is recorded courses. Or if you want to enroll in the test series, you can use IFAS75 promo code and get benefited from the course, okay. Okay, so just now I remember I had given a homework to you. Has everyone solved the question? Yes, yesterday I had given a homework to you. Has everyone solved it? Sir, I don't remember actually, but I will do now, okay. So those who don't remember, okay, about the homework, yes, you can do it now. Just go on to the yesterday's discussion, okay, and just skip to the last point that we were discussing where we have given a homework question for you, okay. Just see this question, okay, and try to answer that in the comment box, okay. Try to answer that in comment box, okay. So now today's homework, okay, today's homework, what you do is you tell me about hemocyanin, okay. Here first thing that you can say is regarding the stretching frequency of oxygen, okay. Don't try to answer here guys, okay. Try to answer this in the comment box. The second thing regarding color, okay, the second thing is regarding color of your oxyhemocyanin, okay. I am majorly discussing oxyhemocyanin, okay. And yes, these two things, if you can predict it, it will be very good, okay. Okay, so it was very nice having you with us, and it was a very wonderful session. Hope you guys enjoyed the session as well as got to learn so much things, okay. So take care everyone, be happy and study, okay, learn things, okay, be good in your preparation, okay. And with this good note I would like to end this session. Thank you so much guys, thank you so much, and happy learning to all of you. Thank you so much guys, thank you so much. डिजिटल वाला कॉन्सेप्ट बहुत अच्छा था सर, थैंक यू सो मच, थैंक यू सर. Yes, thank you so much Mamta for valuing the concept, for understanding the efforts that we put, okay. And for more such concept, but only you need to stay connected with us, okay. So let's meet tomorrow sharp 9 a.m. only at your one and only lovely channel, that is IFAS Chemistry YouTube channel. Okay, thank you so much.